Sir, can we start now?
Yeah.
Great. Okay. Good morning, everyone. Welcome to a new episode of BITS-TEC Innovation Series that spans across various aspects of innovation management, along with the technology development journey of innovators through technology case studies on design, development, and deployment of innovations. In the current episode, we are exploring the theme Navigating the Regulatory Landscape from Concept to Compliances, focusing on understanding the complex regulatory challenges and how to adeptly navigate through them with effective compliance management. Today, we will learn how to tackle such challenges and manage the compliances with the insight from our speakers, Dr. Subrahmanyam Vangala, founder of Reagene Innovations, and Dr. Akash Chaurasia, the associate professor from BITS Pilani Hyderabad campus and the co-founder of Atravis Healthcare Private Limited. Before we begin, I would like to request Mr. Rajneesh Kumar, Head of Tech Transfer Office BITS Pilani and co-coordinator of BITS-TEC, to elaborate on the initiative by DST.
Thank you, Achal. Good morning, everyone, and I thank you for your participation today in today's innovation series itself. And I will also like to welcome our speakers, Dr. Subrahmanyam Vangala and Dr. Akash Chaurasia, for conducting these two sessions specifically curated on pharmaceutical industries and for academic institutions and departments working in pharmaceutical sciences. Just to elaborate on BITS Technology Enabling Center, this is one of the programs of Department of Science and Technology. Under this scheme, 22 institutions have been awarded this scheme, and BITS Pilani is one of the institutions who is a recipient of Technology Enabling Center program. And we have been entrusted with the responsibility of a state of Rajasthan. The mandate of this program is to spur innovation across the region of Rajasthan by creating a platform and by bringing different stakeholders from the ecosystem that involves academic institutions, startup companies, MSMEs, venture capitalists, and other ecosystem players who are working in the area of innovation and promoting innovation to come on the common platform and discuss the various gaps and address them by conducting various curated programs. Innovation Series is one of them. In addition to that, we conduct various other programs like roundtable conferences. We did two of them around renewable energy and for the textile industry for the region of Rajasthan. In addition to this, we do a lot of workshops and training programs for academic institutions and MSMEs for IP training to make them enable to understand the importance of IP and how they can protect their IP. We also train the manpower there itself to create an office of intellectual property that can help the institution and maybe nearby institutions around it. The whole idea is to create innovations out of the academic research, connect them to industries and the regional MSMEs, sorry, and take those innovations into the market itself for deployment through these enablers like SMEs and startup companies. Today's workshop totally focuses on the quality management related aspects in the pharmaceutical industries from academic system, which Professor Vangala will going to cover today. Professor Akash Chaurasia in the second session will try to cover the different innovative products and what are the challenges and gaps that an entrepreneur and a startup company from an academic system faces while the development of these products require a different set of arrangement, and how the partnership in the ecosystem can help you take these innovations into the market itself. With this, I just hand over back to Achal again for the introduction of our first speaker and a brief about the program.
Sure. Thank you, sir. So let me introduce you to the first speaker of the day. Being a seasoned pharmaceutical expert with research experience in biochemistry and molecular toxicology of research, Dr. Subrahmanyam Vangala joins us with 30 years of experience. Even in the pharmaceutical industry, Dr. Vangala has held leadership roles at major companies like Pfizer, Purdue Pharma, J&J, and Shire in the USA, as well as multiple CROs in India. Currently, Dr. Vangala is the CEO and founder of Reagene Biosciences, a translational research company that modernizes drug discovery through innovative technologies like 3D bioprinting, microfluidics, and multi organ on a chip models. In today's event, Dr. Vangla will be elaborating on the topic regulatory compliances by research and development laboratories, good laboratory practices with quality compliance and quality assurance. Welcome to the event, sir.
Yeah, thank you for the introduction, Anshul and Rajneesh. Thank you, Rajneesh, for inviting me on this platform to give my viewpoints on my pharmaceutical industry experience and the journey that started almost 30 years ago, where I was very naive biochemist coming out of academia and trying to adjust into the pharma industry, which is a different ball game, totally different ball game. And that actually emphasized uh, a lot of things that were not emphasized in the academia, like at Berkeley or uh, in University of Colorado, or where I did my PhD in Canada. Uh, you do a lot of innovation, research, great publications with a lot of emphasis on great innovation and everything. But at the end of the day, any work, if it is not, however innovative it is, if it is not reproducible in other laboratories, that data is useless, totally useless. Because you need a global reproducibility of any data that you generate anywhere, whether it's academia or industry. I mean, the other day I was watching some person, I mean, a professor, who was almost ready to be Nobel uh, nominated for a Nobel Prize, and some people could not reproduce his data, some other laboratories, and his nomination was withdrawn. So that's the catch. I mean, you go up to the Nobel laureate, Nobel Prize nominations, and your Nobel Prize nomination was withdrawn because your data is not reproducible elsewhere. And that is where the quality is so important in either academic research or industrial research. So I'll put something that I learned for the last 30 years, why I had to comply with a lot of different things. I'll give you what people call it as a total quality management, TQM. This I heard the word back in 1993. This is very critical that without the quality management systems, the you cannot pass through the FDA approvals of any drug that is gone through the clinical trials or IND packages. So, what is total quality management? In a general definition, okay, in a general definition, total quality management is a standard management practice wherein an employee within an organization continually analyzes its processes to improve the quality of R&D products and services. That's a broad general definition. What are the advantages of it? Enhance customer satisfaction. If I'm a CRO company trying to do some work for a company that uh, outsources the work to me, but they are the ones who submit the package to the regulatory agencies, uh, it depends on my quality. If FDA is not satisfied with my quality, my customer is not going to be satisfied. So you need to provide packages to my customers to get the customer satisfaction, successful compliance to regulatory guidelines and audits. So this requires a lot of regulatory knowledge and how agencies like FDA operate and audit the facilities and improve the reproducibility of research. As I said, if you cannot reproduce the research quality at any corner of the globe, then it's going to be a not a great work that you've done. Implementation process, how do you implement these quality management system? Conducting frequent management employee training to identify and remove problem areas in business and audit. Even if you learn about FDA audits or customer audits or anything, but within your own organization itself, you have to have systems and you have to constantly monitor your own quality management and also provide training to all the employees and identify problem areas and remove those problem areas for successful business and R&D operations. So you probably have seen this many times. You're talking about IP, right? So if I take 10,000 to 20,000 candidate drugs, I won't call them drugs because they're not marketed. So they're out of these candidate drugs, it goes through the funnel for one drug to make it to the market. So even if I patent all this for file IP for all these 10,000 candidate drugs, there's only one IP that really matters at the end of the day because that's the only one that goes through the market. So the other 9,999 IPs are totally are in the garbage can. Unfortunately, that's the success rate in pharma industry today. Uh, why the drugs fail? I'm not going to talk here that, but clinical trials are the basic major bottleneck, uh, where we actually go into the phase one trials, and by the time we come out of the phase three trials and submit an NDA for FDA, 90% drugs fail in the process, either due to the marketing problems or drug synthesis problems or toxicology problems or efficacy problem. Drug doesn't work for what it is indicated for. So there are lots of reasons why clinical trials fail. And only one drug goes to the market. You're talking about $4 to $6 billion are spent. For example, it depends on the therapeutic area. If you take Alzheimer's, neurodegenerative diseases, there's hardly any molecules there. So even if you spend $6 billion, you still don't get a molecule out there into the market. Whereas if you take the anti-infectives, probably you can get a drug within $2 billion. So it depends on the therapeutic area you're looking at, which therapeutic area is more successful than the area. So in the whole process, I'm going to take you to the quality management processes during drug discovery and development. Early drug discovery, they're just screening processes. You don't really need to pay attention to too much of quality management, but yes, it's still important there at that time because if you get very bad compounds into your drug development programs, so your drug discovery was a waste. So there are three at least major practices that regulatory agencies uh, mandate the laboratories on. Good laboratory practices. This is primarily a preclinical area. If I am doing animal trials for efficacy or animal trials for toxicity assessments, these have to be conducted according to the FDA regulations called the GLP regulations. The next comes is the good clinical practices. Any drug that goes into the clinic, it has to be safe, right? So if it's not safe, then you may kill the patients. So that's why preclinical GLP practices are very important, followed with GCP practices in the clinical drug development. More than these, you also are very important in manufacturing practices. You probably heard very recently that many drugs that are manufactured probably doesn't comply with the processes. So for example, some people say, hey, there are counterfeit drugs that are manufactured. These counterfeit drugs don't have a drug in them, or they have a lot of impurities in them, but they still make it to the supply chain. You can go and buy the pharmacy stores. You don't even know whether the drug is actually inside. So these are all counterfeit drugs that are probably mimicking Aurobindo, Sun Pharma, Ready Labs packages. Some people are doing it to cut corners and get these substandard medicines into the market. So manufacturing practices are very, very, very critical because you got to kill people if these manufacturing practices are not adhered, not only during the clinical trials, but after the drug is approved into the market, you need good manufacturing practices to comply with. So how do you manage all this quality compliances? If I'm the CEO of a company or a managing director or a president of the company, I have to put in systems in place. If I don't put the systems in place, I'll be the one who's going to be the loser at the end of the day. The major system that I put in is quality assurance. This quality assurance team must know the regulatory norms. What is the regulatory is expecting? And these guys have to meet and talk to everybody in the district. For example, uh, for a chief executive officer, the direct reports includes R&D lab heads, clinical research head, manufacturing head, human resources head, finance and marketing head, etc., etc. Also project management head. But even the quality assurance has to be reported to the top of the line. They cannot be reporting to me if I'm only the R&D head. I cannot have quality assurance head report to me because I can manipulate the quality assurance head and ask him to overlook some of the quality compliance issues. So the quality assurance head must report to the head of the institute, not to the R&D heads or human resources or finance and project management. And this is very critical process. So if you look at it here, quality assurance uh, deals with R&D heads. If I have 5 R&D heads as a CEO, each R&D head will be assigned somebody to work with through the quality assurance with me. Same thing with the clinical research, manufacturing process, marketing and finance, project management, etc. So these are all dotted line reports, not direct reports. So all the people who are reporting, my teams are reporting into this quality assurance, are only reported through the dotted lines, not directly to the quality assurance. So that way you actually can control your science better. And the quality assurance is the one that will take the whip and say, hey, you haven't complied with this, and they'll go and complain to the whoever is responsible to uh, bring the system back into normal. For example, chief executive officer is responsible to ensure everything is going according to the uh, proposed quality management system. Quality assurance group is responsible for institutional standards. What they have to do is first put together a standard operating procedures. These are operating procedures through the regulatory defined protocols, all equipment, instrumentation, software, adherence to timelines. All these things, SOPs have to be defined. And issuance of laboratory notebook to scientists. Each and every scientist for a particular project has to be assigned a new book. That book comes from quality assurance. I cannot just go and buy a laboratory notebook and start writing my own data on it. So that has to come from quality assurance notebook. Once a notebook is finished, completed, that notebook has to be handed over to the quality assurance group who will archive that in an archival area. Issuance of laboratory notebooks to scientists. Same thing. Protocol preparation by study directors. Study directors must first prepare a protocol which has to be reviewed by the quality assurance teams, and of course the scientists themselves too, and the timely execution of all studies. If I'm saying in my protocol, yeah, I start today my study, and my study completion, including the report writing, will be done today, say August 30th. So, hey, by December 31st, I'll have signed report available. That must be complied. Even that timeline has to be complied. You can say, oh, I have other things to do, so I'll postpone for another month or two. It's not acceptable. So the timeline compliance is very, very important in the process. So prioritization is so important in such cases. So you need to say, hey, I'll take 10 projects, but you're not going to complete any of those 10 projects in 4 months. So which means you don't know how to prioritize your own work. And the prioritization is very, very critical in these cases. Archives, all protocols, study reports, SOPs, SOP training, raw data, electronic data, wet data. Wet data means sometimes I take tissues for histopathology. And they also need to be stored somewhere. And wet data archives, employee job descriptions. Each and every employee working in the GLP area or quality management systems must have their job description defined completely. Being a toxicologist, I cannot just go and work in manufacturing studies unless I'm trained and my job description allows me to do so. So employee training to the records. Each and every employee must be trained with the SOPs and other procedures that these records are under the purview of quality assurance group. They are the ones who will document all this information and archive all this information. So what is test facility management? I'm a CEO. I'm responsible for ensuring the processes are in place. For example, I'll designate the quality assurance team uh, to come up with a program, quality assurance program for any particular project or anything. This quality assurance personnel will not work on the project. They are only overseeing the project to ensure you're complying with the regulations. They are not allowed to work in the project in your projects. Test facility management should ensure that there is a quality assurance program with designated personnel and assure that the quality assurance program is being performed in accordance with the principles of GLP. I'm talking about GLP here, so I'm talking about GLP. But there are other things like good manufacturing practices and good clinical practices are also there. So I have to ensure that all these principles, regulatory principles, are followed. For example, if I'm conducting a GLP toxicology study, a study director has to be named first. And his name is responsible. He has to conduct all in-life toxicology studies along with his support staff. Assign study pathologist responsible for a pathology report. Assign bioanalytical scientist responsible for bioanalysis report. Assign toxicokineticist responsible for toxicokinetic report. Project management team responsible for ensuring timelines are adhered by study director, individual investigators, and study personnel, and they report that to quality assurance team as well. Um, Here, without this kind of a system in place, uh, a toxicologist cannot just go and do the study at his own will, and it's going to be very difficult to sell that kind of a study to FDA. So what is the quality assurance team responsibilities here, right? Quality assurance, there are lots of them here. I'm not going to go through everything here. Quality assurance team from time to time within their own organization have to do internal audits or internal inspections. That is very, very, very critical. And they have to have approved study plans, SOPs. If they SOPs also which will have a timelines. For example, if I write a SOP for an instrument today, typically we do it give an expiry date of 2 years, before which if there are any changes to that protocol, we need to redocument it and re-go through the SOP amendments and reissue the new SOP after the end of the before the end of the 2 years expiry date. So compliance with the principles of GXPs is very, very important in all inspections. As I said, the frequent in-study QA audits have to be there. There are three types of inspections in this case. Study-based inspection, they only first look at all the study and make sure that everything is done according to the plans. And facility-based inspections. Facility means sometimes my facilities may not have the right conditions, right? I mean, I don't have good, what do you call the germ-free system. If I have an animal laboratory, right? Animal laboratories to be germ-free. It's not just a study material. If not, my animals will be contaminated with the germs, and that will alter the outcome of the study. So facility inspections are also very important to ensure. At one time, one company came to a company that wanted to, FDA came to audit. I'm not going to name the company. And apparently, this company was burning the documents so that the FDA won't know what mistakes they did. So FDA actually revoked their license, GLP license. So you cannot just go and burn the documents at your will and say, hey, I'll rewrite the documents. When FDA comes, I'll show you the written document. It doesn't work that way. Your documents have to be thorough from the start of the experiment to the finish of the experiment. And all these documents have to be under QA supervision and have to be mandated by each and every employee. So process-based inspection. Sometimes the processes are very important. As I said, if I'm doing toxicology study, right? There are at least four components: a pathology component, bioanalysis component, toxicity component, and other components as in the software components. All these things are process driven. So to ensure that all these systems are in place is called process inspection. So the data is the most critical here. And if I didn't document it, the data documentation was not done. According to the FDA, even if you've done a Nobel Prize winning study, it's not done. That's it. They should be able to retrieve the data and audit the data and to ensure that it has been complied to the FDA regulations. So don't think that I can write the data tomorrow or day after when I feel like doing it. No, it has to be done parallel to the experiment. All the data has to be documented. Documentation, documentation, documentation. This is three do's very important of the in pharma industry. Quality assurance must find balance. They should not overdo it. Sometimes some quality assurance people start overdoing it, and that's not good for the company either. Evaluating the study type and critical phases. As I said, toxicology has 4 phases in order to achieve a well-supportive view of the GLP compliance at the test facility on which the study is conducted. So the final report, when it is issued, that confirm the methods, procedures, and observations are accurately and completely described, that the reported results accurately, completely reflect the raw data of the studies as well. So it's also very important that the raw data that you generate, write it up For example, today, most of it is electronic. If I weigh a drug on a weighing machine, now it's in the cloud. I don't have to write that information. The only thing is I have to have my own password to open that instrument. So the instrument knows who is actually weighing the substance and how much did he weigh, and everything has to go into the system. So it's a computer. If tomorrow QA department have to retrieve it, they'll retrieve it through the cloud. So even those systems have to be in place. You cannot say, hey, today my software that I've been using, the vendor of the software has issued a new version of it. Then I have to buy the new version and get it installed. Otherwise, the old version will not be compliant with the new methodologies and everything. So you have to have compliance even to the new software versions, and you have to keep buying. That's why it becomes too expensive sometimes. If I have to buy a software $100,000 every year, updated software, I'm not going to be able to do business because of that. Compliance work is needed. Okay. Uh, what is the study director responsibilities? For every study, there is a study director assigned by the quality assurance team. And this quality assurance team will take the protocol written by the study director and look at the study plan. And during the study course, sometimes there may be amendments to the protocol because, hey, you need to change some of the procedure. Those amendments need to be documented as well. And those amendments, study plan, will be communicated to the quality assurance team before you even use the amendment. Sometimes if I have to amend, I have to immediately go to the quality assurance team, hey, I have to amend the protocol because there's some change here. And that amendment need to be approved by quality assurance team. Ensures the SOPs are followed, assess the impact of any SOP deviations, and takes appropriate corrective and preventive action. If I deviate from the existing SOP, and if I did not document the deviation, that is a major problem. Some people have gone to jail for it by the FDA, came and arrested them and took them and put them in the jail because these guys didn't follow the uh, SOP deviations laws. Ensure that the raw data are documented, recorded, including all errors. See, it's not just raw data documentation. If you made a small error, for example, you said you pipetted 100 microliters or 100 milligrams of the drug you put into the system, but you probably weighed it wrong, maybe 10 milligrams. And then you realize that, oh, I only weighed 10 milligrams, not 100 milligrams later. Could be a genuine error, and sometimes human errors are also possible. But if you must fudge the data and say, hey, I weighed only 100 milligrams because you don't want any quality assurance guys come on yelling at you for making the mistake, then that is a violation of the SOP deviations and other things. So you basically need to document even the errors that you put. So if the errors are not documented, if I'm writing a lab notebook today, right, I would typically at the end of the day, I take my lab notebook for that day, that pages, whatever I wrote. My supervisor will actually review the data and he'll ask me questions why there are some errors here, why there are not so errors there. He'll ask a lot of questions. Only if he's satisfied, he'll co-sign it. Otherwise, he will not sign it on my notebook. And that this is essential for even for quality assurance. Then say, where is your boss's signature? There's no boss's signature, so we're not going to accept your raw data file. So computer systems have to be validated. There's several other, it's called Part 11 compliance. Uh, GLP studies are under Part 58 compliance. Computerized systems are under Part 11 compliance. I'm not going to go into those details. Um, all the equipment used in the study is properly validated and qualified before the start of the study. We have standard processes. For example, if I have a method that I'm validating it, every time I'm using the same method, I need to ensure my calibration curve is exactly the same calibration curve that I'm getting it before I use it repeated times. So I cannot use calibration curve from the past experiments and use that for the new experiment. Every time I need to revalidate the calibration curve. Sign and date the final report to indicate acceptance of responsibility. You are showing that you are actually, I'm a study director. According to my knowledge and everything, everything is done perfectly to the regulations, and I accept my responsibility for the protocol and the report issuance on this one. Now the study personnel, people who actually do the work are the ones sometimes make the mistakes. So they need to be trained, knowledge of the GLP principles or GMP principles or GCP principles, access to the study plan and appropriate SOPs. They need to know what the SOPs are required for that particular study they are conducting, and they have to be trained on them. Must be periodically trained on SOPs and comply with the instructions of the SOPs. Record all raw data. They must record everything into the notebook, including any errors, as I already pointed out. Study personnel are responsible for the quality of their own data. If you make a mess and the entire study is compromised, which took 6 months and maybe $2 million to complete, you are the one who is responsible for failing the entire study. So be careful and ensure the quality of the data that you present. No fudging, no overwriting errors or anything. Exercise health precautions to minimize risks. Sometimes we handle dangerous chemicals or chemicals vapors, things like that. So you need to have personal protective equipment to wear. For example, if I'm working in the lab, I used to have steel-toed shoes. So something I don't understand in India in many places, what they do is, no, don't wear shoes, don't wear slippers into the, even doctors won't let you wear shoes and slippers. I don't know whether they are ignorant about the safety issues, or I have no idea why. Sometimes I see that. You must wear shoes that are resistant to the acids or any heavy, for example, something fell on your legs, right? You should not wear chappals in the lab. You should not wear any slip-ons, and you should not wear silk clothes. There are laboratory rules you have to follow and maintain that so that you are not injuring yourself during the study process. So that will also ensure the integrity of your own health, your own safety, and the entire study. So I'm not going to go into this one. There are lots of, I'm giving you this presentation. So if you want to go look at this OECD Environment, Health and Safety publications, series on principles of good laboratory practices and compliance monitoring. They also list a lot of good laboratory practices and GLP data integrity stuff. Anybody can go through it. So which are the regulatory agencies that I deal with mostly? US FDA, OECD, or if I submit our INDs and NDAs to Europe, European Medicines Agencies. In India, CDSCO, Central Drug Standards Controls Organization. And ICH is something very important to understand. If I have a new protocol that I think is a very, very novel method that I discovered, and I want this to be applied in my GLP study, first, you cannot just use them in the GLP study unless we show that it is a validated process. I'll give you one example here in that International Conference on Harmonization has taken the toxicology protocols from pharmaceutical companies, Environmental Protection Agency, and environmental toxicologists, and many other places. What they do is they've created a standardized, harmonized protocol. So if I do the study here in India with that protocol, if somebody does this same study in another country with the same protocol, the data should match. If it doesn't match, that means the protocol is not validated internationally. So today, the international validation of the protocols is very, very critical in taking any new method. We are doing these 3D bioprinting methods or microfluidic methods, right? These are still not under regulatory thing. But if I have to get them validated, I'll say, hey, first, I need to validate in different laboratories. I'm a company that started it, who will directly submit this data to the FDA pharma companies. So I'll go to the pharma companies and say, hey, pharma companies, I have this new method protocol. Can you validate it? If they are interested in incorporating that protocol into their own system, for example, I can get some money from them. Say, hey, $1 million. So 5, 6 companies come together and wanted to validate this new methodology protocol. They will pay me $1 million each. And my data will, not just the data, my samples will go to all these 6 companies. All the 6 companies use the same method and see if the same data comes through all these 6 companies. Once that is established, they will go to the FDA and say, hey, we have a new method protocol that is validated according to the regulatory confinements. Can you approve this as a new guidance? So they'll take it to the guidance and everything. So I participated in several of those FDA town hall meetings in the previously for metabolite testing in animals or drug-drug interactions in animal in vitro systems like that. Several protocols that I had to go and meet with along with other companies to define the protocol validation. So FDA comes up with a guidance at the end of the day. So that is called International Center for Harmonization. You need to harmonize. Even if my company, that probably I have one division in Bangalore, another division in Pune, third division in Delhi, all these 3 sites must have matching data when we repeat the study at all these 3 locations. So here's another one here. You can see that all these OECD series compliance monitor, lots of these GLP protocols are issued by OECD. Same protocols will also be found in FDA as well. So let me go back to say, why did this happen? The creation of GLPs. What is the historical perspective here? In the 1970s, FDA became aware of cases of poor laboratory practice all over the United States. So they decided to do an in-depth investigation of 40 toxicology labs. When they went and audited the labs, and they found a lot of people are fraudulently, even without conducting the study, they're writing up the data, and poor laboratory practices. Then after the completion of a series of hearings in 1975, 76, and 77, US Congress proposed and then enacted the Good Laboratory Regulations for FDA 21 CFR Part 58, Good Laboratory Practices for Non-Clinical Studies. This is for GLP. Of course, We have other GMP, GCP also came along later on. The proposed regulations for good laboratory practices were published in the Federal Register on November 19, 1976. The good laboratory practice regulations final rule was published in the Federal Register. For example, if I have to do a toxicology study, there's a rule that I must conduct in 2 species, one rodent, typically Sprague Dawley rat, and one large animal, either dog or monkey. Dog, it is a beagle dog, or monkey, if it is cynomolgus monkey. So these are very, very regulatory defined animal models that I need to use. I cannot just go and use any other animal model. Instead of using Sprague Dawley rats, I cannot just go and use basic C3F1 mice or Wistar rats or something else. So these are FDA documented. If you want to change the animal model, then you have to justify to the FDA why you need to do it. You can say, hey, my pharmacology is better in this particular model compared to the other model. You need to justify it to the FDA to ensure the FDA accepts your data in another strain or species. Okay. The proposed regulations for good laboratory practice were published in Federal Register. Okay. In December 22, 1978 is where the final rules were published. Now, OECD principle. OECD is nothing but GLP principles are also, but this is, there are lots of OECD countries like European countries, etc., use more OECD norms. India is actually comes under OECD GLP principles. So these are also issued in 1978 and revised in 1997. All these principles are actually constantly being revised based on the new data the FDA sees, and they'll try to issue more rules into the system. So US CDER, Center for Drug Research, often sees compliance statements citing utilization of the OECD principles on GLP. So here is one FDA gentleman, Zhu Chen, published it in the web, and he gives some directions on how to, I think it's 2022, sorry, July 19, 2022. I'm going to go through his presentation a little bit here. So what is FDA GLP compliance inspection program? According to him, the FDA staff verifies safety data and support the form tox review process. The quality and integrity of non-clinical studies used to protect human subject safety. So what happens is that if I have very poor data from the animal toxicology studies, the doses that I select in humans may be wrong doses. In such case, I can go with the wrong doses into the human clinical trials and compromise the human safety. So this is very pivotal for the GLP toxicology studies to ensure that we protect human subjects with a conservative dose that we go. For example, if I do rat and dog, right? My drug is toxic at 400 milligram per kilogram dose in dog, for example. So I'm not defining what toxicity it is, but in my rodent, a rat or something, it may be safe only up to 100 milligram per kilogram. There's a 4 times difference. So now, according to the FDA, I cannot use the dog data. I have to use rat data because it's most sensitive species. Based on that, I have to use the pharmacokinetic, toxicokinetic data and everything, come up with the allometric scaling data. There are formulas available by FDA, use it, and come up with, and I also know the pharmacology dose. If my pharmacology dose is only 10 milligram per kilogram, but I cannot use that dose in the clinic. So I have to come with maybe less than 1 milligram dose for a 70 kilogram human, not 1 milligram per kilogram dose, 1 milligram per. So this is a conservative estimate we use to define how do I design a clinical trial. So you don't want to compromise the human safety when I do those calculations wrong. For example, if my drug is toxic at 100 milligram per kilogram, but you probably used only 10 milligrams with error somewhere. I made documentation error, and I thought it was only 10 milligram per kilogram. So what happens is I'm still thinking that it is safe up to 100 milligram per kilogram. So to ensure that when you do go to the clinical trial, you're not going to kill patients or human subjects. You need to make sure that you have very highly conserved dose go into the. It doesn't have to be necessarily a pharmacology dose in the humans. So we submit INDs, NDA, new drug application. ANDA means ANDA is for generics mostly. Okay. BLA is biological license application. These are mostly for antibodies, biological drugs, etc. So GLP inspection program by FDA, the animal rule studies, as I said, pivotal animal efficacy studies need to be conducted in the relevant pharmacology species and animal PK studies. Typically, I do 4 animals in pharmacokinetics. Mouse, which is only 20 grams size, whereas rat is about 200 gram size, monkey is about 4 kilogram size, and the dog is about 10 kilogram size. Now you compare to scale it up to 70 kilogram human, average 70 kilogram. So we do a lot of different studies here, animal studies, and we try to predict what can happen in terms of the pharmacokinetics in the humans. If we don't get the right pharmacokinetics, we also need to know whether the animals have a different drug metabolism compared to humans. And based on that, we also do some in vitro animal drug metabolism studies to ensure that we correct for the differences in animals to humans. Based on the correction factors, we go with the particular human dose that we do that. So the animal rule studies is very, very critical in selecting the right doses. So natural history studies sometimes not inspected, but there are also directed inspections and surveillance inspections are present in GLP. What are directed inspections? Address concerns about data integrity and study conduct. Review division's concerns. What is division's concerns? FDA's concerns. Confirm GLP. Yes, I did everything GLP. If they're not satisfied that you've done under GLP, they'll invalidate your entire study. Confirm data integrity, reliability of pivotal studies. What we do is, as I said, if I do a GLP toxicology study, before that I do a dose ranger pivotal study, a non-GLP study, to ensure that I'm selecting the right doses to go into the pivotal animal. The GLP toxicology studies. So if there are any differences, then I have to do corrective actions since last inspection. And OECD study audit. OECD can come and audit my studies. Some people, as I said, they failed the audits and their GLP license was suspended. Complaint follow-up, data falsification, which is a serious violation. People went to jail for falsifying the data. Lack of GLP compliance. For FDA center participation, FDA comes and participates. No predetermined number of testing facility inspections. They say, hey, you're only going to do 1 time or 2 times. No, they'll come any number of times they want if they're not satisfied with the data. At the end of the day, they're looking at the safety of the patient, of the humans. There are surveillance inspections. So as I said, there are things that when you, one of the things I want to hear emphasize is something called a master schedule. when an FDA comes in, let me go down to what are the documents they typically ask for you. Organizational chart, floor plan, master schedule of studies. What studies are you doing? Sorry about it. SOP index, all the SOPs list, equipment index, and all these equipment or have SOPs and they're validated. What are their calibration schedules? If I said, according to my SOP, I calibrated my instrument today, and according to my SOP, within 3 months, by end of December, I need to do another calibration. If I don't do that, and if I use that instrument for another study, because it's not calibrated at the prescribed time at the end of the December, FDA will not accept the data. They will say that this is violation of the SOP. Study director, study personnel, training files. If I am part of a study, my training files for SOP training, study training, everything, my CV, my expertise, all has to be in the training files. These training files every year has to be updated, and the quality assurance group will actually archive the updated files. Pest controls. For example, I mean, when you're using animal studies, you don't want pests in that area. So you need to make sure that all the pests are not present at the facility. FDA inspections typically last 5 days. When FDA comes here to inspect my labs, the inspector will conduct a facility tour and interview personnel. The inspector will review many records, including study-specific documentation, data, facility and equipment records, standard operating procedures, training files, etc. So what are the focus areas for FDA inspection include, but not necessarily limited to, but this is the standard list I'm talk about. Test and control article, if I'm making a drug to dose into the animals, right? I need the drug manufactured. For example, if I have to do a toxicology study, I cannot use 50% impure material. At least a minimum 95% purity study I need. But when I go to the clinical trials, I have to have 99.9% pure material to do the clinical trial. So 95%, why we use it in toxicology studies, if there are any toxic impurities in the drug material, they will also be qualified that they are not causing any toxicity in the same toxicology studies. So test and control. For example, if I am taking a new drug that I want to test whether it's toxic or not, but I need to make sure that there is a positive control, validated control that is also used along with my validated control should be giving me the historical data that we already have on files, mimicking exactly the historical data. So you need to make sure formulations are very important. Most formulations must be generally recognized as safe formulations. If I take an exotic formulation today, hey, I'll use any formulation because my drug is soluble and better than that. No, you cannot do that. You have to use only FDA recommended, generally recognized as safe uh, formulations. Test facility management. They'll also look at what are the management qualifications. Are they really knows how to run the studies? Study director responsible. If the study directors are qualified or not, study personnel are qualified or not, and what is the quality assurance unit responsibility? Are they qualified or not? Because they are the ones who are putting the systems in place, quality systems in place. So study conduct, they are the ones who are looking at study conduct. So final study report, including contribution to scientist reports, and they also, it has to be archival records. So they look at archives. First thing is they'll just go and look at the archival room. They will come and say, hey, I want to go and see your archival facility areas. So then they'll look at the facilities, equipment, SOPs, and everything. What are your procedures for the animal care? And because today you cannot just use any animal at your will anywhere, because there are lots of animal ethics regulations as well. You have to follow those animal ethics regulations. And electronic data capture system. How are you capturing your electronic data? What are the softwares used? Are they validated softwares? A lot of things. All these things FDA will look through it. So it all after 5 days of inspection, if inspectors will hold a closeout. I've been to many of those closeout meetings before. If no deficiencies are found, everything FDA was happy with, and then there's no Form FDA 483 is issued. If Form 4 FDA 483 is given to management, listing the deficiencies found during the inspection, some of them may be minor, but FDA still highlights them. Those findings can be corrected during the inspection process itself, but these findings will still be listed on Form FDA 483. This FDA 483 document is for public. I can go and look at anybody's 483 on FDA website if I want to. So those are public knowledge, what happened in that particular company. So remember, the GLP is a quality system that ensures the non-clinical safety studies are appropriately designed, conducted, and documented to generate reliable and reproducible data anywhere in the world. So that's, in essence, what I'm talking about is how do you manage it. So these quality management systems not only relevant to pharma industries, but are also relevant to diagnostics companies, medical device companies, even academia today. So people must be on their toes. So many people say, hey, I hate this documentation. I don't want to do this. But there's no choice. If you're into this system, you have to follow the guidelines. Okay. Thank you very much. Hello?
Yes, sir. So thanks so much. I think we can take few questions in case we have.
So
Look.
we have a question in the chat box from Sarvesh. He's asking, sir, how can we ensure the accuracy and integrity of these documents?
That is why you have quality assurance team. Quality assurance team is responsible to ensure that accuracy and integrity is part of the study conduct. So you have SOPs, standard operating procedures, well-validated methods, and everything that has to be. So it's a system, that quality management system comes through the quality assurance teams, and which will put rules, ground rules. This is the way to do it. That is where you ensure the accuracy and integrity of the studies. Without quality assurance teams, you can never do it. As I said, I can just go and work in my silos in the corner of the lab, and I do something and nobody reviewed it, then that data is not valid for me to submit it to the FDA.
Sure. Dr. Vangala, I have one question. Let's take another question from one of our participants. Achal.
So, sir, it's a follow-up question. So, he wants to know, can technology like digital documentation systems improve the process?
Well, it's still not ready yet. Digital documentations are still evolving. For example, people are talking about artificial intelligence and everything. They're still not ready in terms of the because these are all new processes, new methods, and everything. They're not validated. For example, you're talking about uh, digital clinical trials. People are talking about today. What are digital clinical trials? If somebody is going to do a clinical trial on a new drug on me, before they do clinical trial on me, they'll do a digital twin on me. So they'll have my genomics data available, my proteomics data available, my transcriptomics data available. So a virtual, my Subrahmanyam Vangala will be created, and the clinical trial will be conducted on the virtual Subrahmanyam Vangala before they actually select me to directly dose drug and do it. But that systems are still not ready yet because you don't even know how much. For example, the other day I was reading about the genomics data. Uh, so far, there's about 250,000 European descent people genome sequencing is done. Out of those 250,000 people, there are about some 250 million sequence mutations. Imagine we have 8 billion people in the world. The data is only represents 250,000 people. So by the time you get the 8 billion people data and put it into all this digital system and come up with validated methods and everything to do your digital clinical trial, you're going to go probably another decade. We still need to do it. So digital systems are not ready into the regulatory compliance yet. There are some cases there are, but not all cases. In many cases, especially experimental science, you need to do this study. For example, a lot of people are doing artificial intelligence at very early stage. But that early stage artificial intelligence cannot let you say, hey, my drug is safe. It, it works in an artificial intelligence system. But I cannot just go and directly go to the clinical trial without doing an animal trials. I still need to do the animal trials to prove that the drug is safe, right? So if I have a drug that is mutagenic, causes genotoxicity in Ames test, oh, usually I don't take it into the clinical trials because I don't want to take a chance with somebody, a patient or human getting a cancer or something. So I won't do it. But if my drug is not mutagenic in that system, I cannot assume it won't cause cancer in humans. So I still need to do animal trials before I go to the clinical trials. So you have to, the digitization at this stage, not ready yet in terms of the clinical trials and making it easier for us to get new drugs into the market faster and easier. So I think we have got a long way to go.
Sure. One more question, Dr. Vangala. Are these total quality management practices country specific?
No, as I said, they are not because today we are international. Everything is international. For example, if I have to do a clinical trial in US, right? The genome populations in the US are different from India. In such case, if I have to do a clinical trial in India, I have to do the genome sequencing of the patients to ensure that they have the same genome sequences for the same target, and I can design the clinical trials. But if I don't have that data, I cannot just. So India does allow the clinical trials for a drug that is approved in the West. But again, no, the genome is different for India versus the Caucasians, Indians versus the Caucasians. So you really need to be very careful in how to design. So if I have to do a toxicology study, this format is the same globally. I cannot use my own format for India separately compared to Europe or US or Japan or anybody. The International Center for Harmonization dictates that I have to use the same system, quality management system, throughout the world. That's it.
So there is a follow-up statement. How strange it is that some documents will be responsible for public safety like TQM in pharmaceuticals. What is your opinion?
Sorry, again, repeat.
How strange it is that some documents will be responsible for public safety like TQM in pharmaceuticals. What is your
Yeah,
opinion?
my opinion, it is critical for safety, the human safety. So I don't want to give a drug without safety established in animals to humans, right? So I am doing research on saying that animals may not be relevant to humans, but that's different because I have not proved it yet that using my 3D bioprinting or organoid systems or anything, I have not proved it. They're relevant to the human systems compared to animals. I can keep saying that animals are not relevant to humans, but FDA is not going to buy into my say, okay, keep doing your work and show us the data. If you are satisfied tomorrow that your models are also clinically relevant, human relevant, then we may use those models for clinical systems. So you need total quality management, even for the new methods that I'm developing, like 3D bioprinting, organoids, spheroids, microfluidics, everything. That needs to be validated through the total quality management system.
Sure, sir. Thank you so much for your presentation.
You know,
And
Thank you, Dr. Vangala, for such a nice presentation and covering some of the practical aspects of quality management in pharmaceutical industry. And since your experience comes directly from the industry itself, I'm sure that the participants must have today have understood the nitty-gritties that they need to follow once they are working on their innovative products, and when they are following and when they reach at this stage where they have to follow these standards in order to qualify their innovation for as a marketable product. So thank you so much for your presence, and we appreciate your time and effort to have this session with us today. Thank you so
Thank
much.
you. So, I do say that you guys should have a quality assurance team in your own premises so that they will ensure the quality, high quality.
Sure.
Okay.
Yeah. Thank you. Thank you, Dr. Vangala.
Thank you. Okay. So I can sign off? Or-
Yes, in case you want to stay back for the next session, you are most welcome to join us.
Yeah, I'll stay back. Okay, no problem.
Sure, sure. Yeah. Thank you so much.
okay. So move.
Dr. Akash,
Let me remove my presentation.
Yes.
yeah, please.
Okay. Okay.
So moving ahead, we have our next speaker in-house. Dr. Akash Chaurasia is an accomplished researcher with extensive experience in pharmaceutical R&D, having worked at leading companies like Dabur Research Foundation, Dr. Reddy's Laboratory, and Amneal Pharmaceuticals. His expertise lies in the formulation development of complex products such as liposomes, nanosuspensions, and lyophilized products for regulated markets like the US and EU. Currently, Dr. Chaurasia is an associate professor at the Department of Pharmacy, BITS-TEC Hyderabad campus, where he leads research on developing complex injectables in collaboration with pharmaceutical industries. Being a BIRAC BIG awardee, Dr. Chaurasia also has co-founded Atrevis Healthcare Private Limited, a startup focused on developing innovative and affordable healthcare products. In today's event, Dr. Chaurasia will be talking about his experience on the development and translation of the innovative pharmaceutical products. Welcome to the event, Dr. Chaurasia.
Dr. Akash, are we audible to you?
I'm audible?
Yes, yes, we can hear you.
Okay. If I'm audible, you can see me also.
Yes,
Thank
please.
you. So, please see the screen also. Can you see my screen?
Yes, yes.
Okay. So, thanks everyone for joining this session and you for inviting.
Dr. Akash, your voice is quite feeble. I think the mic is a bit away from your end, or maybe you can adjust it a bit.
Okay. So now voice is fine? Or you want to increase?
A bit better, but not so clear.
Just a minute.
Dr. Vangala, are you around?
Yeah, I'm here. I'm here.
Okay, great. So while Dr. Akash rejoins, there is a last question for you. So somebody asked, what are the consequences of non-compliance with documentation standards?
First, you will get your product.
So I'm audible now. It seems like my voice is clear.
Yes, Dr.
Yes.
Akash.
Yes, Dr. Akash, in the meantime, we have just taken a question and Dr. Vangala is just responding to that.
Yeah, I mean, there are two things that if you don't comply, you're not publishing papers here. You know, in
Can
industry,
you hear me?
yeah. Okay, yeah. Here's my answer. See,
Yes, sir.
I cannot hear actually. So
if somebody doesn't comply with the, I mean, in academia, a lot of people do publish papers. So they don't do regulatory work. Basically, they say, hey, I published 10 papers, 20 papers, 30 papers, and so they count the numbers. Okay. Most of them are peer reviewed by the experts in the same field. Does it mean that that data can be submitted to the FDA? No, it cannot be. Because the FDA looks at quality compliance. Without quality compliance, publications are no use. Or so you must have a quality compliance, total quality management to ensure that your data will be reproduced anywhere in the world. It's the must. Okay, so non-compliance means either you lose, you won't be able to market your product, or regulatory will say, hey, we're not going to approve your product into the market. So if you really want a product into the market, follow the regulatory guidelines. That's it. Non-compliance will not help you.
So non-compliance will basically add additional challenges for your commercialization.
Yeah, absolutely. Absolutely. Correct.
Okay, great.
Can you ask Dr. Akash if he is able to listen to us?
Sure. Thank you so much, sir.
Yeah.
Meanwhile, Professor Akash, are you able to hear us?
okay. So
Are you
good
able to hear
morning
us
all.
now?
It seems I'm audible to you all. So I will be starting now. Just give
Sure,
me a
sure.
quick yes if you can see me, you can hear me, and I'm just sharing the screen also. Okay, thank you. So it seems you can see the screen also. Just a minute. Okay. So yeah, myself Akash. I'm working in the Department of Pharmacy, BITS-TEC Hyderabad campus as a faculty. Today I'm going to speak about development and translation of innovative pharmaceutical products. Okay. So already something has been told about me, so I will not be going into all those things in detail. This is just what I'm doing right now. So I've done my pharmacy in a bachelor, master's, and PhD from the various institutes. And basically, I'm a trained formulator. I'm a product development scientist. I work with various companies, starting my career with Dower as a research trainee. And my last assignment in the pharmaceutical industry was with Amrich Pharmaceuticals in Ahmedabad, where I was leading a team for complex injectables, primarily the liposomes. Here I'm working as associate professor in BITS planning. Recently, I founded a company also along with my PhD. It's called Activist Healthcare Private Limited. I'm also in the board of studies for Parental Drug Association, the association which is meant for providing the guidances for the manufacturing of parental products, injectables, vaccines, and all. Okay, so this is all about what I'm doing right now. So coming to the topic of our discussion today. So basically, today's discussion is primarily meant for the pharmaceutical products, basically the innovations in the pharmaceutical product. So this will be divided in two parts. First, I will go through just run through some of the basics of what things needed for translating a pharmaceutical product. In the second part, which possibly I will be demonstrating a more on the case study, specifically on a product which is meant for us and which was our first product of our company. So I will just show you through run through the journey of how that product, so that can give you a real time example of how the things were done in that particular case. Before starting the things, just have a quick overview of the difference between an invention and an innovation. Being an academic scientist or being working in an academic labs and all, we are more sensitive toward doing something new, something more into an invention mode actually. But it is equally important that we can translate that kind of invention into a particular product actually, and that is how innovations comes into picture actually. So invention is something creation of some kind of new product or the processes for the first time, some kind of principles, theories also sometimes. And once we are converting that process or the product into some form which can be useful for the society or for the people actually, that is what the innovation is all about. So basically, it is an improvement of existing improvement in the existing inventions or any kind of significant change. And if we look into the pharmaceutical space, all kind of pharmaceutical products and all, you will find there are so many of the innovative products lying around. The basic principles can be relatable in those things. For example, if, let's say about most of us know about, let's say, liposomes. It's a lipid-based carrier system. Nowadays, so much of in use in the vaccines also, mRNA-based deliveries, lipidic nano carriers and all. So somebody has came out with the concept of liposomes long back, around 60 years back from now. But for a specific product, we can add on a value into that system by showing the utility of a particular pharmaceutical product, and that can be a kind of an innovative product that itself looking like a very simple way of doing the thing, but it is not that simple. It's very difficult. Just I was listening to the previous speaker, and that was really great knowing about all those things actually, whatever the clinical things are needed, and each and every product is special actually. Okay, we cannot generalize the product just based on their category and all. Each and every product is having their own requirement, having their own studies which are supposed to be done. Same is the case here also. So each and every innovative product is supposed to be taken up very seriously in a specific manner only. And that is what the difference between an invention and an innovation is all about actually. So now the important question is why and when to innovate. Nowadays, so much of talks are going around like we should do something innovative, we should do this and that. But I try to find out the things like in what scenario we should do that actually. because it is not a cup of tea for everyone. Actually, it is something you should be passionate enough to do all such kind of thing. So there are other kind of properties which are also required, or characteristics are required in case if you're really looking for innovating a product actually. So you should be passionate enough for your job, that kind of work actually you're looking forward. Any kind of innovation, either it's a pharmaceuticals or it's a biopharmaceuticals, biologics, or any other kind of or any other kind of product you're working in. You should have a creative mind in doing so, because so much of principles, so much of inventions are lying around. Now we need to connect the dot. We need to come out in the form of some products actually, and that is what the creative mind can do in such cases. Eager to do something different, obviously. Understanding the problem statement is very important, like because that is what the basic difference between doing a research in the basic science and doing a research in the application base actually. Because in the application, when it comes for, we need to identify what kind of problem is really there actually. Many times the problem is looks very simple, but the solutions can be more difficult actually. Okay, so none of the problem is simple. We need to identify, we need to go to the root cause of the problem actually, and that is how the solutions can come into. So that is very important to understand the problem statement. We'll go through the case study in our case, and there I will show you how the problem statement can be identified, just relating with our product. Ready to take on the challenges which are associated. Challenges can be of any type. It can be a technical challenges, non-technical challenges. So we should be ready for all those things actually, if we really want to work in the area of innovations actually. We should have something translational invention which can be converted into a product actually. That is very, very important. It should not be a me too kind of product which others are also doing and then it will of no use. And the most important thing is not for the sake of fulfilling any kind of requirement, kind of academic, government, or bureaucratic requirements actually, because nowadays so many things are happening around, and like so many kind of push are also there in the entire ecosystem also. So that is very, very important, that if we are doing something, that should be coming out because of above, just a minute, because of the above reasons, not for this reason actually. That is very, very important, and that is where we should innovate actually. We should not be doing it in that case. Possibly it will look very easy to do some innovation, to have our own company and startup and all those things, but the journey is going to be very tough in all the cases. So please be very aware of what you really want to do. You need to identify the way you want to process actually. So not for the sake of certain specific or materialistic requirement where which is not going to land up into a real thing actually. So now coming back to the things like how we can translate a product from an idea to the market actually. So that is very important. So what is the journey of a pharmaceutical or a healthcare product actually? So everything starts with an ideation. Ideation can be of any kind. It can be of simple just a idea which can be good, which can be general idea also. The important thing is we need to work on that idea at the early stages only. We need to do all the possible ways to refine that idea by doing the literature review, by reaching out to the people, by getting some experience in that particular case. We need to relate that idea with the market and the societal need. That is how we are. If we have to go for a market, it should be of some value. Otherwise, it will be of no use. So it should have some market or societal need. Then only it will make sense. So whatever the idea is coming out, how it can be positioned into the market actually, that is very important. So ideation to the market need is very, very important at the early stages only. We need to do that by any of the way. Nowadays things are relatively more easier. If I go back and look into the things 20 years back or 10 years back, now we have an access to so much of the information just on the click of our hand also. And obviously we need to do some extra effort also in doing such thing. So once we have identified the idea, once we have identified the market need or the societal need, then we need to work on generating the proof of concept, like how whether this idea is really working or not. So since right now I'm talking about the pharmaceutical product, so we may need to do some initial POC work where we need to prove that, okay, if we are looking for a pharmaceutical product, the most important thing for a pharmaceutical product is it should be safe and effective. So if we are really working for an extremely new kind of molecule, then the journey is going to be very long. In case if we are looking for a reformulation kind of work, then we need to work, already the clinical effectiveness or the safety has been established. Maybe we need to do some bridging study at the later stages. But at the early stages, we need to look it from the perspective that the product should be stable. That is the most important part for any pharmaceutical product. It should be stable actually. So such kind of initial work to demonstrate the safety, to demonstrate the stability, to demonstrate the efficacy. Using some initial tools can be of use. Here we can do the in vitro studies to prove the safety and efficacy of the product. Here we can do some preliminary stability studies to see the stability of the developed product and all. So there can be any ways where we can demonstrate the effectiveness or the application of the product by doing the POC work. So once this proof of concept is generated and we are getting enough confidence, because this POC work has to be done for our confidence, that is very, very important. So we have to be very truthful on what we are doing actually. Once this POC work is done, once we have validated our once not validated, once we have just got an initial proof of what we are looking forward is really doable or not. Then it is a time for the prototype development. So prototype development is a product development basically. So product development, there is a various ways depending on the nature of a product. If it is for the parental application, then the development could be in a different way. It is for the oral application, it can be in a different way. If it is a biologic product, then it will be in a different way. So we need to, based on the nature of a product, we need to work in the area of development. That is what comes under primarily in the core R&D activities actually. So once the prototype product is developed, supporting activities are also established like analytical studies and all. IP landscaping is already done. In such cases, we need to go for establishing the safety or efficacy before moving into the humans. So what is needed in that case is the preclinical studies. The purpose of the preclinical study in a GLP environment is to just to establish whether the drug or that particular product or that particular, whether it's a new product or it's a reformulation work or repurposing work, it is supposed to be safe for the human consumption or for the human studies actually. And that is the purpose of preclinical studies. So we need to do preclinical studies that can again be divided into two parts. First can be for generating enough data for our confidence, can be a non-GLP based study. But for the regulatory requirement, for the regulatory filing purpose, for the regulatory from the regulatory bodies perspective, they need your preclinical study should be done in a GLP environment. And why I'm bifurcating this non-GLP and GLP is because the level of seriousness, the level of investment goes up to manifold when it comes for the GLP based studies actually. And that is what is always expected by any regulatory agency. Just I was going through one question where somebody has asked about the non-compliance. If somebody is not following the compliance, there is no way out for that. Okay, so there is no way out if you're not following the compliance, if you're not fulfilling the work of as per the expectation of the regulators actually, then it is whatever the kind of work, whatever the novel work you are doing, it is of no use actually. So it has to be done as per the, because regulatory agencies has established various systems to do that. Okay, so all those GLP, GMP, GCP comes under that system for their reliance only. So preclinical studies are supposed to be done for demonstrating the toxicological part, primarily whether the drug is toxic or not. Based on this preclinical studies and possibly the manufacturing or the development activities done at that particular stage, we need to go for the IND filing, investigational new drug filing activities, IND, and that is primarily comprised of animal pharmacological data plus the manufacturing information plus the clinical studies protocols actually. So then we need to go for the further level of studies for the scale up and validation so that we can supply the material for the clinical studies actually. So based on all these informations, based on the preclinical data which we are having initially can be of non-GLP nature, but later on we need to generate the data in the GLP environment. We may need to do the scale up and validation studies. The most important thing is before moving into the highly expensive, highly time consuming clinical studies, it is always advisable to have some kind of meetings to understand the perspective of the agencies actually. So in such cases, nowadays, all the good regulatory agencies, whether it is US FDA, EMA and all, they have a system where they can accept the request for the pre-submission meetings and all. That pre-submission meetings can be pre-IND meetings, pre-ND meetings, pre-AND meetings actually. So in this pre-submission meetings, we can come up with our queries, specifically the technical queries, not any kind of general information and all those kind of filing related information. That should be very technical information. We can come up and we can ask the FDA, suppose in case if we are using some new kind of excipient, some new kind of component, or we are using the drug for some other kind of indication, some other route of administration, then what is the understanding, what is the requirement that can be there from the regulatory side. So having such kind of meeting will be really helpful to fine tune our studies also, whether we are whatever the protocols we have developed, whatever the studies we are planning for the next level, that all we can understand at this stage. We can also negotiate with the regulators also. If we have sufficient data from other sources and all, we can negotiate. We can also get some kind of waive off also for doing certain type of studies. So pre-submission meetings is not mandatory. It is not mandatory, but it is highly useful. It is not a part of a regulatory system. Okay. It is not something which is compulsory is supposed to be done, but it is highly mandatory. This will be helpful for any kind of innovator company or any kind of innovator product to reach to the market on time with the minimum investment, with the minimum effort, because this kind of meeting will be helpful to validate our thought also that whatever we are planning to do next can be really helpful. So once based on the pre-submission meetings, whatever the clinical studies or toxicological studies are required, that are supposed to be done. Okay. And again, this clinical toxicological studies are going to take a good amount of time in doing. It takes many years also in doing so. And once we have enough data available which demonstrate that the product is safe and effective and obviously stable also, then there will be a time for the regulatory submission. Whatever the market we are looking for, if we are looking for a global filing, we need to go with that strategy. If we are looking for a country specific filing, then we need to go accordingly. We need to plan our regulatory submissions actually. Okay. For example, for the US FDA, the possible filing pathway for the new product is NDA, either by 505B1 approach or 505B2 approach. That they are the two approaches. B1 is something, it's a new chemical entity. We need to go for 505B1. 505B is basically a paragraph of FDC Act actually. So, and if we are going for some kind of reformulation or repurposing, then 505BT approach is there. Same is the case with the EMA also. In case of EMA, we have a various kind of filing systems like centralized system, decentralized system. So all based on the strategy of an innovator, which market they're planning to target, which country. In Europe, where they have EMA has more than 30 plus countries actually. So they have a different kind of filing system. But US is very clear with their filing route, that is NDA. Para 505B1 and B2. And for the generic filing, there is another system, ANDA system, which comes under 505J, para 1, 2, 3, 4 filing. So based on your product, whether it's a NDA product, B1 kind of product, B2 kind of product, or it is a generic product, accordingly the filing and the regulatory submissions are supposed to be done. So this was all about how the product can be translated from one stage to the from the idea to the final stage actually, and obviously such kind of product require so much of patience and so much of time duration and the funding actually. Just for the information of the audience actually, the filing fees for the NDA with the clinical studies is 40 CR, 40 crores in INR somewhere. It's in dollar. Just I converted it into crores for this year. For 2024-25, the filing fees is only the filing fees, nothing else. Okay, the filing fees is 40 crores. With if you are submitting something with the clinical studies, if you are not going with the clinical studies, in that case they will give you 50% discount. So it will be 20 crores. That is only the filing fees, and rest of the things are required for the clinical studies. That is much more higher than what the filing fees is. So it is going to be a very long journey with so much of investments are needed at the various stages. So we need to be ready in all such cases. Anyway, so coming to the just a minute, I will just see if any query is there. I believe we will be taking up the questions at the end only. Okay, so maybe. So coming to the case study in during COVID outbreak actually. We suffered a lot. We suffered a lot with various kind of non-availability of the drug. One of the drug was remdesivir for injection. It's a lyophilized product. Lyophilization itself has a limitation with respect to the batch size enhancement and all. It is not easy to enhance the batch size of the lyophilized product. So there was a shortage. There are other reason also, but I will focus more on the technical side. So remdesivir for injection, there was shortage because of such thing. Then immediately after COVID, in the few countries like in our India, we come across one new disease that was a black fungus. Okay, so in case of black fungus also, again, there are very few drugs which are approved for the treatment of black fungus. So broad spectrum antifungal drugs like amphotericin. Now again, amphotericin is also available as a liposomal preparation. It's highly renal toxic actually. So it comes as a liposome. So the drug is entrapped inside the liposomes. So it will be reaching to the specific site of action to the fungal side and doing the needful. Otherwise, it is highly toxic toward the kidney. So it available only as a liposomal product or in the lipid complex actually. It is not available in the general form where the drug is simply solubilized in water. That is very dangerous to give to the patients. So, and which is very expensive. And again, this liposome again is a lifeless product. So first we need to make a liposomes and then we need to do a lyophilization of a liposome. Now, in the normal lyophilization or a freeze drying, it takes around 2, 3, 4 days for the lyophilization process. Again, that requires some wisely designed lyo recipe. But in case of liposomal product, the lyophilization itself is very tricky. So we need to go very gently. So for the, to best of my knowledge, for the lyophilization of the amphotericin B liposome will take around 6 to 7 days. So if you're manufacturing a batch today, let's say today is first of the day, then the product will only be completed by 7th or 8th of the month. And then it will go for the QC release. Since it is for the injectable purpose, sterility test will take 14 days again. So the product which you have started on first of the month is going to be released possibly by 21st to 25th only. It will take 20 to 25 days for this particular product to get released because of the complexity involved in such system. And that was also one of the reasons for the non-availability. And there are many drugs which are available. I'm not sure how much how they were entrapped properly in the liposome thing and all, because it's not easy to make a liposome itself, and then again lyophilization increase the issues. Another drug which was used for the purpose was the posaconazole actually. So this was the drug which brought our attention also actually. So we have identified the problem actually. It was not only black fungus, there are so many other issues also. So this drug, it is used for, it's a broad spectrum antifungal drug used for the various kind of fungal infections actually across the globe. Okay, and there's a very severe occurrence of the fungal infections in India also. Okay, so serious fungal infections which lead to hospitalization is more than 4% actually. And obviously during COVID outbreak also black fungus was also one of the indication for this drug actually. So there are very fungal infection apart from causing it can be a matter of death and life also, but in the normal scenarios it can be a kind of a social stigma because of this fungus infection. The patient will really feel so much of difficulties in their social life also. So finding a right antifungal drug with a broad spectrum activity is very important. Now the problem with this drug is this is a drug again which is innovated in USA. MSD is a company which has came out with this drug. There are few generic players also available, but the costing of the drug is very high. Per tablet cost is around 1,000 rupees. The duration of therapy in case of various kind of fungal infection is from 6 weeks to 12 weeks. And the drug viability is also very low. So per day the dose is around 600 to 800 mg, and the tablet you can see on the label also the tablet comes as a 100 mg tablet. So means initially you will be needing like 6 to 8 tablets as a loading dose, and then as a maintenance dose on the daily basis you will be needing 3 to 4 tablets. So the costing for any kind of fungal infection using this broad spectrum antifungal drug is somewhere around 1.5 lakhs if you go with this Noxafil innovator product, because of the low viability. And the high dose is required because of this viability issues, which again lead to various kind of serious side effects, and obviously the fungal resistance also. And obviously this all lead to high cost of treatment, high toxicities, high dose requirement, and completely it is not patient compliant actually. Okay, patient compliant is not only something which we are giving to the patient and they are very easy to administer. That is not the only case of patient compliance. Patient compliance also associated with the side effects or associated with the after effect of the drugs actually. And nowadays all these antimicrobial resistance is really going to cause us so much of issues in the future. So these are the various areas we have identified, with the problems we have identified at that stage actually. And that is how we come out with our solution. We come out with the product which is supposed to be novel, uh, because we came out with a product which is patentable and we have filed the patent. Also feasible, it is really feasible, because many times the novelty is inversely proportional to the feasibility. It has been observed many times, especially when we are doing something in an academic research actually. So to make novelty with the feasibility, sometimes simplicity has to be added upon, and we bring the simplicity factor. Then many times people will not take it very seriously for you, because normally for getting a good academic project, for getting a good academic work, some kind of complexity should always be there. Then only we'll get a good publications and all those. But that is not a right thought actually. That is absolutely not a right thought. If you're working on a simple solution, if you're working on a feasible solution, people will appreciate as good as if you're working on a complex solutions also. So please, if you're having that kind of understanding, please come out from that. You do the simple things, you do the things in a proper manner, you will be able to go for the publication with a good publication. You will be able to convert into a good product also. That is very, very important. So we come out with a solution which is novel, which is feasible, which is simple, and which is having a good commercial value. And this is what our product is all about. So the product was a tablet. We come out with a different kind of formulation which can be given as a capsule actually. So this was a novel formulation which is stable. We have done the stability up to 6 months. We have done the stability, the improved doses regimen because of the increased viability, which we have already proven at the later stages actually. So this all we have done actually. So the novelty of the product is like it is stable at room temperature. Uh, it is a novel formulation. From the mechanism side, it based on some nanotechnology kind of work, which is really easy to absorb through the system. Okay, enhanced oral viability, which we have already proven through some initial preclinical studies. Simple manufacturing, as I told you about the issues with respect to the manufacturing, it's simple mixing, which can be mixed together in a normal GMP environment. And the scaling up can also be done in a very easier manner. So it is easy to scale as per the GMP compliance, which can be easily done. The machines which are used for the manufacturing are the commonly used machines, normally the jacketed vessels, the normal filling machines and all. They are not something extraordinary is required in such cases actually. So from the feasibility perspective, it was a simple manufacturing process, can be processed in a commonly available mixing tank also. And the results which were coming out is it's going to be a very cost effective, easy to manufacture as per GMP compliance, faster production. Faster production, why? Because to address the issues whenever any kind of pandemic or epidemic situations are happening, then only the importance of all those faster production or easy to scale up kind of things are really required actually. So the benefit which was coming out possibly is a high quality, affordable treatment for the Indian population and then rest of the population across the globe also, which is our next target. Rapid supply in the emergency situations because of the easy manufacturing, easy to scale up, and a high throughput actually. So with all those type of novel thought and all, another important thing is you always look for the business opportunity. What kind of business opportunities? So that we have done some kind of studies at our end. We went through the antifungal drug market share across the globe. When we found the percentage which is there in the Africa, Latin America, North America, Europe, Asia Pacific. Right now we are targeting India, so we are more concerned. We are more concerned about the Asia Pacific. So what we found is India has a good market share for any kind of antifungal infection, and obviously we know that, but from the number side, because numbers are also important actually. So from the number side also we have found the same. So there was a huge business opportunity for our novel drug product. And the another important thing is our drug, basically our entire thing was related to the mission of Government of India nowadays. Government of India is supporting so much of the schemes actually, especially Department of Pharmaceuticals, Ministry of Chemical and Fertilizers is coming out various kind of schemes. They're promoting Make in India, Innovate India, Startup India kind of thing. So all those kind of recently strengthening of pharmaceutical industry scheme also came out actually. So considering all those kind of advantages, the ecosystem which was created actually. So we found that our product is useful for the society. It is in line with the Government of India initiative also, and it is having a very good business opportunity also. So the reduced cost of our medication will be helpful for the Government of India to provide the treatment to all section of the society. So based on this, we have identified the value proposition, which is an important component if you're developing any kind of product actually. So value proposition is nothing, it is the requirement of a customer and how your product is going to solve that customer requirement. So what kind of requirement are there? So patients need something which is really good with the lower cost for the treatment of fungal infection, because the pain they are getting is exhaustive therapeutic regimen, high cost treatment, side effects, high drug resistance, product shortages and all. And the gain they were expecting is the cure and quality of life after treatment actually. That is very important. And the ease of treatment. So what we come out from our product in the form of a gain creator or the pain reliever is effective treatment, quality of life post treatment. We try to address all those issues with a better therapeutic regimen, cost effectiveness, low side effect, and basically because of this lower dose and all, kind of a curve on the drug resistance also, and the availability because of the easy manufacturing thing and all. So understanding the customer requirement, offering the product is what the value proposition is all about, and we have studied that part well actually. So based on all this, under our product proposition, the value proposition, and all, we identified the differentiation of our proposed solution actually. So our proposed solution is basically in line with the Make in India scheme of India, which with the kind of a proprietorship at our end and the import substitution. Because of all this importing thing and all, the costing of the drugs are very high in India. Because in India, it is very painful to say that innovations are not happening to that level as it is happening, because one of the possible reason is the reward is not there for the innovators actually in India. So if you go and talk to the people, the pharmaceutical industries, they are all kind of innovation. We are completely relying on the innovative product from the West actually. Once they developed in the West, NDAs are filed or centralized filings are there. After a long time, they come to India because of many reasons. I will not be discussing all those things here. Most of us know those who are in connect with the pharma industry, all such kind of things actually. But somebody has to bring the change actually. So people need to come out with the thought of bringing innovations in India. Second differentiation was the lean manufacturing with the existing facilities. The third differentiation was it aligns with the government of India schemes actually, Jan Aushi, because we are going to offer this product at a lower price than what the innovator product is. Improved stability for the logistic part, and obviously it will align with the international mission of curbing the antifungal or microbial drug resistance. So these are the differentiators for our product, and whenever you're working on any kind of innovator product, you should be aware of what kind of differentiation your product will be bringing in actually. Some kind of market research is also supposed to be done. Okay, so market size has to be identified. So same is the case in our case also. We try to identify the total addressable market, serviceable market, and serviceable obtainable market. That is what the TAM SAM SOM is all about. So total addressable market, antifungal market is very big in India, more than 3,600 crore. The antifungal drug market is, including all kind of antifungal drug, dermatological, systemic and all. Out of this systemic antifungal drug means oral kind of prevention is 1,200 crores of market. And out of which triazole antifungal drug, which our drug comes under the category, is 1,050 crores actually. So we went very conservative with the approach that even if we are able to serve 1 to 2% of the market share for the triazole antifungal drug, still we are able to do at the initial level a business of around 10 to 20 CR actually. So such kind of assessment are always useful whenever we are looking for any kind of product development, because at the end, if you're developing a product, it has to cater the need of a market actually. So you should be aware of what is the market size, what is the attempts and sum of the product actually. Another important thing which requires from translating a product from the innovation to the translating an idea from the innovation to a product is we need to do a competitive landscaping actually. So we have done that also. We came out with we have done the landscaping on the various aspect, technical aspect also, primarily the technical aspect and the clinical aspect. And from that we have identified that we can offer a product at a very reasonable price with a much more advantage than the existing product actually. One of the important reason is this product has been designed internally with us, the patent is lying with us, and it's a novel formulation with a better clinical application. So obviously all those kind of advantages will always lies with us actually. So based on all this landscaping, we have also tried to identify what is going to happen in the next 5 years actually. So antifungal market, I'm only talking about India right now. Okay, obviously sky is the limit. We can go out of India. We can go serve the need of the global scenario also. But right now, this entire thing was done primarily for the Indian purpose actually. So the rate at which the cumulative annual growth rate for this product is 10.5% in the next 5 years actually. So based on that, understand the primary reason for this growth, there are various reasons actually. So much of government schemes are coming up in the form of Ayushman Bharat and all. And there's so much of investment is happening in the pharmaceutical R&D. So much of recently, like couple of days before, one new biotechnology policy also came into, which is basically primarily meant for helping out the Indian innovators or the Indian manufacturers actually. Rising disposable incomes from the patient side, because they want to people are ready to pay good money also if they really want to come out from the disease, and rise in awareness about the healthy lifestyle possibly after COVID. So based on all those parameters, basically we have identified the various kind of positioning which can be there. So based on all these things, there are a few other questions which possibly we need to answer actually, like when we are going to set up the things, and also we come out with the our company establishment in September 2023 with this idea. This idea was initially generated in the lab in BITS planning, and then after I translated this idea to my company actually address, for which we took the licensing of my own patent for this company actually. So a few other things you should always be doing it, like whether you are going to discuss with the target customer. So in our case, since it is a pharmaceutical product, our target customer will be the doctors primarily, the healthcare professionals. We should have a, so we have done the same kind of things with them also. We reached out to the various kind of people who are dealing with this, and we got a very good positive feedback from that actually. And another important thing is whenever you're translating anything from the idea to the, you should have a good mentors actually, especially the business mentors. So it is good to identify your business mentors or the technical mentors. So we have those people also, we talk to them, and they also help us in making our go-to-market strategy, how we'll be moving actually. So this is what our go-to-market strategy is actually. So we have done this green color of work is already we have done so far. POC work, we have done the prototype, we have done the IP filing. Initial stability was done. Initial preclinical, I will say non-GLP only because we doesn't have a GLP capability with us. So we have done the PK study on the rodent model actually. Currently, we are in the process of scaling up the product. We are in discussion. We are not discussion. We are in the latest stage of doing the things with the with the CMO contract manufacturing organization. We have identified it's a GLP compliant lab. We are going to do our scale up studies and we'll do the stability studies over there. Possibly next couple of months or by end of the year, we will be looking forward to do that. Then after we will be doing the pharmacokinetic studies on the higher variant of the animals on the beagle dogs actually, and we'll do all those kind of manufacturing testing of the of that product which we have which we are doing the manufacturing in the GMP lab actually. So the stability studies, this all is planned for now. Based on this data, we look forward to discuss with the regulators in India actually, and based on, because this drug has already been approved in India for the similar kind of indication. So what kind of bridging studies may be required for that from the toxicological perspective, from the clinical perspective, if we need to do some BAB studies, because that kind of clarity possibly can only be obtained once we talk to the regulators actually. There's so much of guidance is available, but not specific to this case. So we need to have some kind of pre-meetings with them. So that will be the next future of activities we will be doing with. And once all those kind of human studies are done, if required, if we do that, then we will be moving forward for the launching of the products and all. So this is what the milestone is right now that already I discussed actually. And the possible target customer in our case, we may be looking from B2B, B2C and B2G also, like from us to government also, because this is a kind of the problem with the basic health system. So we'll also try to attract the government agencies also in such cases. So that we'll decide at the later stage. So coming to the team, any great work can only be possible if you are having a good team with you. So the core team is me. I'm working here in BITS-TEC and my PhD scholar. Both of us are the founder of this company. Okay, and then we have advisors and mentors from the various section of the pharmaceutical industry. We have an advisor from the as an industry expert. Mr. Mithun Saha is R&D head for the Division Pharma. Then Dr. Saurav Arora, he's a owner of Arbor Pharmaceuticals. He's also a M.Pharm PhD and he's having a very good business skills. He's running this company for last more than 20 years. Then Dr. Ramesh Penama Reddy, he's a regulatory expert for us. He is having his own company, Masu Global Solution. They provide the regulatory solutions for at the global scale. And then we have a clinical expert also, Dr. Prashant Kumar, who deals with all kind of he's an oral surgeon primarily, and he also takes care of he took care of the black fungus related issues and other fungal infection during his tenure. So bringing the right mentor and advisor is also equally important to sharpen up your ideas and your concept actually. So to sum up the things what we discussed, we need to identify the unmet market need. We need to have a good team to do the things. We need to identify if your solution is simple, but that's fine, but it should be efficient. That is more important actually. The scalability we always look into. We should have a clarity on the roadmap. Many things are not clear even with the regulators also. There's no one line rules and regulation in the pharmaceutical industry. One thing you should be very clear actually. Pharmaceutical regulators can give their opinion based on the nature of a product. For one product, obviously there are the guidelines. And if you go and read the guidelines from the ICH, from the FDA also, they never say that this guideline is compulsory for you to follow. They always say this is what the opinion of the agency is. Now, if you have something other to say, you can go and discuss with them. You provide your justification, and then after you can get the thing done also. So it is always needed whenever you are working on any kind of innovative solutions. You will not be getting the answer for all the question at the initial stages. Many times the answer will be coming up at the later stages also, especially with the regulations and all. So please do not stop your work at the early stages just if you're not having an answer for the question. You will be able to find the answer. Many times you need to ask the questions to the people, then only you will get an answer. Quality development, just now, in the previous talk, our speaker was talking about the quality systems and all. That is very, very important. Maybe the objective in the academic research is different from what industry always look into, but still, if you really want to translate the things from one place to another, quality development is always required, including the data integrity. And finding the right partners and funding agencies is very, very important. And that is what the institute like BITS-TEC is always help their people, their faculties and student in reaching out to the right people. We have a TBI system, we have a tech transfer division, and they always provide help in all such cases actually. So I'm thankful to the BIG Biotech Ignition Grant, which is provided for this product to do the things. Whatever I shown that it's ongoing is all because of the support of BIG. I'm also thankful to TBI, Technology Business Incubation Hyderabad campus, Dr. Sandeep Rao, who was with us throughout our journey from the early stages to now. So I'm thankful to all, to BITS-TEC obviously. This is my research group, a bit of older picture, but yeah, fine. This is my lab, this is my BITS lab, and this is my team. For any kind of queries, you can ask now, and you can also find me on my emails and on the web page. So I'm done with this discussion. If you have any query, you can ask. Okay, so just a minute. I need to work with the speaker thing because my speaker is not working. Just give me a second. Actually.
Sure, please. Professor Akash, can you hear us?
Yeah, I can. I'm audible?
Okay. Yes, you are. So one of the questions, it's more of a comment, but maybe we can provide some constructive thoughts around it. So the question is, our institutions are more equipped for teaching and research, and we produce workers rather than innovators and entrepreneurs. So, what do you think? What are the interventions that academic institutions can look into with respect to the syllabus, let's say very specifically to pharmaceutical sciences, that help them nurture new ideas and explore possibilities of entrepreneurship while they are still studying in the 4-year course of their pharmacy or master's course once they reach into M.Pharm?
So, yeah, it's a good question. I can share my experience. The only teaching experience I'm having with me is BITS planning actually. Not having any kind of experience with any other kind of institute, what they're doing and all. I'll just take an example of BITS. So in the BITS, what we are doing right now is we are promoting so much of through various kind of schemes, whether at the undergrad level only or at the high levels also to the faculty, various kind of schemes. Second thing is yeah, it's absolutely right. The courses which we are having right now, we need to follow certain councils of also, like so council syllabus also. So those syllabus, as per my opinion, is not that great actually. So here in BITS also we do some kind of we offer some kind of courses which are really useful for the students. So some kind of tweaking you can always do with the institutes also are not saying exactly you need to follow. Any institute is really interested in providing a quality education which can turn out into, which can be helpful for the needs of a society or the need of the industry. They need to come out from their comfort zone. They need to come out. They need to take some extra effort in crafting. Nobody is stopping them to offer something relevant or something good for their student. So it is up to the institute. Now, obviously, as an individual, we can't change the institute. As an individual, what we can do is we can update our knowledge. Now, as I told you, we have so many other things available with us. Keep yourself updated with the news. Actually, we are associated with a pharma company. Many of us would not have having any kind of exposure with the industry in the past, no need to worry that comes your knowledge in that area. You start reading about the news articles now. You go and access to the regulatory websites, and so much of the information and the things are available. So we can make all those changes at the personal level, the institute level also. We can also stand up. Nobody is stopping us. Blaming the system, I believe the change we do at our end. But we are doing such kind of things even before we started. For example, for this product also, this is not the only product we are working on. We are working on some other product. Just for the case study, I discussed this one. So from the day one when I joined BITS also, very difficult. I can understand the objective of the academic institute is different and the industry is different. We are more looking forward for the number. Industry is not looking for the number. They look for some quality development and all. So somehow we need to find a balance between the things actually. And this is up to us how we can do that. And that is what makes you different from the other also.
Sure. Thank you. I think there's another question, and maybe if you can speak a little bit closer to the mic, that will, I think, improve the voice quality. I just request you, sorry, apologies for that. So another question is, can a small scale product like cosmetic products from the institute lab can come into the market? And what are the regulatory procedures to be followed?
I can give the general answer for this question because neither I am a cosmetic scientist nor I am a regulatory scientist actually. But I can, based on my understanding, I can give. Anything can come out from anywhere. The only thing is we need to follow some quality practices actually. So if you are working at the POC level, fine, you can work in an academic lab. Obviously, you need to do, at the time of POC, you need to do the study which makes sense for that product actually. And when I say sense for the product means I'm talking about the efficacy of the product and the stability of the product. Many times what we do is we always focus on the efficacy part. We never focus on the stability part. Stability is not existing in the academic research actually. It is assumed that whatever we are making, it will be stable. But that is not the case. Many times in the experiment, what we do, we write overnight, we have kept something, and some reaction is happening overnight. It never happens in industry. You can't keep anything overnight. You need to have a control of everything. Okay, so stability is as equal as the efficacy of any product. So when you are working in an academic lab, you are working in the industry, wherever you are working, and if you're really aiming towards converting that product, the important thing is you should look for the efficacy, you should look for the stability. With both the ways you can do the thing. Now, once the POC work is done, you got enough confidence, okay, this product is really working. For the development activities, you will be needing some quality system.
Yeah, this is Dr. Mani Vangala. Just to reiterate it, you see, cosmetic industry is now banned completely the animal studies. In India also, I mean, many countries have done it. India is one among those countries. But it doesn't mean that you can do get away with the quality compliance issues because they're highly regulated under OECD protocols. So if I have to do a cosmetic development, I need to know continuously what are the OECD protocols. For example, cosmetic products for skin versus the eyes. There are tons of OECD protocols available. And if I have to set it up in my lab right now and get them GLP validated, my lab will take 2 years to get the GLP certification to use those protocols. So it's not just easy that yeah, I'll do, it's cosmetic, but especially if you're talking about a new cosmetics that are not out there in the market, then you have a more uphill battle to do it. But most of the cosmetics we are talking about today are formulations, ingredients that you're adding to the old compounds. So it depends. We still need a lot of GLP studies to do that. Okay. And the GLP compliance is very important, and your labs have to be certified as GLP. If your labs are not certified as GLP, you cannot do those studies.
Yeah, it all depends on the regulators actually. Yeah, so
right.
FDA is very serious about all such kind of thing or any regulatory agency. So that product is coming under what kind of regulation. That is also very, very important. Actually, so first of all, we need to identify the market, which market we are targeting actually, and accordingly, the entire development and regulation can be done.
Sure. Yeah. I have one question for you, Professor Kash. As an academic spin-off that you have created at Revis Healthcare based on the intellectual property that you generated in your lab, what are the challenges which a spin-off faces from an academic system when they have to scale up the product? Are those facilities, let's say in terms of facilities, are good enough in the academic institutions, or they need to move out? And what are those kind of collaborations and those kind of ecosystem stakeholders they get into touch with in order to scale up their product from an academic system?
Something which we are facing on the day in, day out. So, yeah, it's very challenging. We have a basic amenities available with us. We have a machine to do the things. But if we need to take it something out, we need a good quality system always. So we may be needing that, and that is what we can do in that spin-off only. That is not possible in the academic system. Creating or maintaining a quality system is not something which everybody will appreciate, because people are having their different objectives while working. Everyone is not looking for developing a product actually. We're dealing with the basic research. Even in the case of new product development also, initial studies are supposed to be done in non-GLP or non-environment also to get some experience. So different people are working in a different area. So we can't come up with one system to do that. For that, what is needed is a separate system which possibly we can do with establishing a company and all. Okay, so we can have our own quality system in doing so. Challenges are there. One of the most important challenges is understanding actually. Understanding the requirement for the pharmaceutical product. So that maturity and understanding is still missing in the entire ecosystem. People are not ready to understand that pharmaceutical products will usually take time. Take many years to come out actually. Many times people compare our product or pharmaceutical product with some kind of application, some kind of devices or diagnostics which are very easy to come related to a pharmaceutical product. Because pharmaceutical products are meant for human consumption, unlike the diagnostic tools, unlike the devices and all, which are not supposed to be meant for the human consumption actually. So we need so much of rigorous toxicological studies, clinical studies, and investment to do that. So we cannot come devices, diagnostic, or any other kind of system are having their own challenges. Regulatory systems are not that advanced for those systems actually, and there are other challenges. So we cannot compare each and everything all together actually. And many times what is happening is people are bringing everything under the same table and all, comparing one thing to the another, which sometimes lead to some kind of confusion for the entrepreneurs or for the innovators. But anyways, the key to do the things is keep your work going, and that's what.
Sure. Thank you, Professor Kash. In case there are no other questions. Yeah.
So I would like to pose a question to both the speakers for the event. So if someone at an individual level would like to read more about the standard operating protocols or the compliances, all the rules and regulations, is there a easily accessible common platform where one can read about all this? If you know.
Yeah, there are lots of places. I mean, you just have to look for it. But I mean, the SOPs are available. I mean, most organizations do post their SOPs online on their quality systems as well. So you have to go and look for it. Yeah, there's a lot of accessible information out there.
Any top two which you can maybe name so that people can start from there and then explore according to their interests?
I believe the participants who are here, they are well aware of ICH. So ICH is something which is the agency which provides the guidances about the various stages of the product development. Basically, the product development is meant for three major things: safety, efficacy, efficacy, and manufacturing. So ICH has a guidance for all the three areas. So people can start based on their interest area. If they are a clinician, they can go for the safety ability guideline. If they are related to the manufacturing part of the development, they can refer to the quality guidelines. So those guidelines are available. Apart from this, US FDA has n number of guidelines available with them. Whether it is about nanoparticles or liposomes or about the novel products or about the biowaivers, there's n number of guidelines available. So it all depends on how to start with actually. So first the person needs to start like where they can start with, because again, coming up with so many guidelines will be very confusing. SOPs is something which many times is something specific to a company. So I believe SOP you meant by all these guidelines only. Those kind of guidelines
Yes,
are
yes.
available in domain at free of cost. Just you need to use Google rightly. ICH Q1 guideline or ICH L1 guideline, just you need to know what guidelines is available, or US FDA particular guideline. I think every month, every time new guidelines come up. Second thing is US FDA, if you go to the US FDA website, they have a learning center. Okay, so teaching and learning center is also, not teaching exactly, learning center for the industry professionals. So they keep on doing the webinars. There's so much of learning programs which are automated actually. You go there, starting from the basics of drug product development and approval thing. You need to go there, you need to just, it's free of cost. You need to go through the presentations, you need to answer few queries, let's say 5 or 10 questions, and then after they will give you the certificate of online only. Like Dr. Akash Chaurasia has completed such and such webinar, something like that. So we need to go and put our efforts in reading all those. People can start from there.
Sure. Thank you so much, sir.
Yeah. Before we close, just I will take another 1 or 2 minutes. As I earlier mentioned that we run all these programs under the support from DST under this scheme of Technology Enabling Center. I would just request our my colleague Sahil to just take you across on the services that we offer to the ecosystem partners, whether these are MSMEs, academic persons, or innovators, startup companies who would like to avail these services under this project from supported by DST. Sahil, over to you.
Yeah, thank you. Thank you very much, sir. As Dr. Vangala and Dr. Akash already emphasized on the importance of regulatory compliances and IP filing, I would just like to touch the pain point that what innovators and scientists they face. So basically, protecting your idea, intellectual property is the most important thing. And as we saw that product will come out, like it will take time. After so many years of the innovation, it will come into the market. And after clearing all of this regulatory compliances, if you see that your product is getting copied by somebody else, it won't be nice, na? So first of all, we have to protect your intellectual creativity, intellectual creation in the form of IP. So we BITS-TEC, supported by DST, we are here to handhold you. If you are stuck in any kind of IP related aspect, you don't know how to file it, what to do with the patentability search, what is trademark search, what are the requirements of filing. So we are here to help you, handhold you throughout the process. And one of the major pain point for the students and faculties is the patentability search, because identifying the novelty and inventive step is the most crucial part of this journey. So you don't know whether novelty and inventive step is there or not. So our patentability search is mandatory. It is most essential before filing patent, before putting your money into that whole process. And if you see in the market, the average patentability search cost something around 25K to 60K to 1 lakh. So it's something which is not feasible for students and faculties. So here we in BITS-TEC we offer a comprehensive like patentability and trademark search, as well as recently we have started also design filing and design search also. and will completely handhold you throughout the process. So if you are facing any kind of IP related issue, IP related query, you need any handholding, please write to us, please connect with us. We'll take you through, we'll guide you through entire process. Thank you, sir.
Thank you, Sahil, for taking across the various services that BITS-TEC offer to our ecosystem partners. With this, I request Achal for the closing remarks.
Thank you everyone for joining us for this event, BITS-TEC Innovation Series. So with everybody on the expertise, we saw how to do, what are the challenges, again, things to do and not to do. So we thank everybody, Professor Vangala, Professor Chaurasia, for joining us and giving us our precious time. With this, we conclude our episode. It was great to see such an interactive audience, and whoever could not join or has a follow-up question after the event, we will help them reach out to you. And with this, we would like to thank all the participants for joining. The participants, if you want to get in touch with any of our featured faculty, inventors, our guest speakers, or even the team members, please feel free to reach out to us. We also welcome any comments or suggestions for the current or the next episodes. Thank you so much.
Dr. Chaurasia, can we get your presentation? If it is not confidential.
You're asking for Professor Akash presentation?
Yeah, right.
Professor Akash?
Yeah, maybe we can connect in person. We'll discuss.
Okay. So Dr. Vangala is asking, can you share your presentation with him?
Can you provide his email ID or contact details? I will talk to him.
Sure,
Yeah.
sure. We'll do that. We will help you facilitate that. So thank you, Dr. Vangala, and thank you, Professor Akash, for your time and efforts in conducting this today's session. I really feel it was really inciting because regulatory landscape is one area which is generally neglected and generally not linked into academic system at an early stage. But this could be a potential bottleneck in case one is scaling up the product from an academic system leading to a market entry. I think now there is a lot of organization support and a lot of government support in terms of early funding that helps innovators, academic innovators to develop the product to a certain level. There are organizations, there are Section 8 companies, not-for-profit companies in the similar area that can help you understand the regulatory pathway. But definitely this is one area that need to be looked into when one is starting product development stage, and they need to address it so that they don't feel any possible hurdles in the near future. If an early stage precautions are taken in terms of product development, that will help you navigate all these regulatory aspects in a more easy manner. So with this, I want to thank our speakers today and all the participants for your interest in attending this workshop. Please do join us for our upcoming workshops and keep visiting our BITS Technology Enabling Center website for new updates and new programs. And in case anyone wants to reach out us to for any of the IP services, we have shared our details. You can directly reach out and write us your request. We'll love to entertain your request and would like to help you in your all the endeavors. Thank you and thanks. Have a good day all.
Bye.
Bye-bye.
Thank you.
Thank you.